morning, everybody. Thank you, Duncan, and the organizer for inviting me to this looks to be a very interesting meeting, as usual. So my talk today is on the on nanobodies. The target that we are working on is the HCN channels, and HCN channels are found. There are 4 subtypes in humans: HCN 1, 2, 3, and 4. They belong to the superfamily of the KV channels, and they share the tetrameric organization. The monomer contains a voltage sensor domain, and in the cytosolic part, a cyclic nucleotide binding domain. The peculiarities are that they are activated by hyperpolarization, and that they have a mixed conductance. They also have only one drug on the market, which is ivabradine, and for cardiac dysfunctions, and approved for cardiac dysfunctions. And it's a open pore blocker that acts from the intracellular side and does not discriminate between the 4 isoforms. So the physiological role of HCN channels is very well was discovered, let's say, in cardiac, where HCN4 is the predominant isoform subtype. And as you see, this is the current that slowly develops in a voltage and time-dependent manner of HCN4. The activation by cyclic AMP that increases the current, but not the maximal current, because it generates a shift in the activation curve. It's a right shift that for HCN is an activatory effect. And the functional role of this channel is shown here in this sinoatrial node action potential, where the current, the native current, which is called IF, contributes to the diastolic depolarization phase with a tiny current, but still is an important effect because the slope of the depolarization is related to the heart rate. So changes in IF change the slope, and they can accelerate or decelerate the heartbeat, which is one of the effects of cyclic AMP, of course. So as I said, HCN are also heavily expressed in other parts of the body. In particular, HCN 1 and 2 are neuronal, and therefore they are related to a large number of important diseases in humans. So defects in HCN4, they lead to sinoatrial node disease like bradykardia, while HCN1 and HCN2, they are discovered to be related to epilepsies by Christelle Depien, and also related to neuropathic pain by the big work of McNaughton. So therefore, there is a need for drugs that can be subtype specific, because of course, if you inhibit HCN2 in neuropathic pain, you have a relief from pain, but you also have problems on heart rate. So you are this is related to the unspecificity of the drug ivabradine. Another problem is also that we realized working on a class of mutation that gives epilepsy, mutation HCN1, is that we found that both gain of function and loss of function mutations lead to the same type of disease, which is this early infantile epileptic encephalopathy. And in fact, we found mutations in patients that when we tested in our in vitro system, which is X cells, lead to gain of function because they have a right shift of the activation curve. And these are mostly located in the voltage sensor domain. Then we found mutations in the pore domain that are leading to this develop of the instantaneous current, which is also a gain of function, classified as gain of function. Then we found mutations in the C-link CMBD, which are related to a left shift in the activation curve. So this is a sort of this is a loss of function mutation. Therefore, the need is for finding also different modalities to interact with the channel. So we need maybe for blockers for the instantaneous, but we also need modulators of the voltage sensing even within the same class, the same pathology, in the same subtype. So in conclusion, we thought that a good idea was to screen for nanobodies. And I don't have to introduce the quality of nanobodies to you. These are camelid nanobodies, camelid derived, and they are in particular they are very small compared to the other antibodies. They have other properties. They are soluble and they are stable. But the smallness is one of the best characteristics because they can enter in crevices within the protein where probably the larger antibody they cannot reach. We think that to obtain subtype-specific nanobody would be useful for instance, to block the pore or to modulate the voltage sensor, but also even if they don't have a functional effect on the channel, they can be used to deliver nanoparticles specifically to the channel. And in this regard, I want to mention that there are no antibody available for HCN that are targeting the protein from the extracellular side, because they mostly are raised against the cytosolic side of the protein. So we decided to screen this library, which was created by Macahon and et al. It's a gift from them. And they created this library of synthetic nanobody, which are displayed on yeast cells. So each cell of yeast expresses at the surface after the cell wall, at the cell wall, a nanobody, and we screen a different nanobody, and we screen it with the purified protein of HCN4, which is connected to a GFP, and then we use GFP to retrieve the yeast cells by magnetic activated cell sorting, and then we were also using FACS at a certain point. So the screening of the library included rounds of enrichment in which we were selecting the cells that were binding to HCN4 in the open pore conformation, and I will explain this in a moment. But also steps of depletion in which we removed all the nanobodies that were recognizing the protein in the closed state pore, and included several rounds, which we in parallel were checking the enrichment of the library by FACS. So we used this screening of alternating enrichment and depletion because we are able to purify the HCN4 protein in two states, with the closed and the open pore. And when we saw the structure, we realized that not only at the pore level there are differences between the closed and the open state at the expected site, which is the bundle crossing. But there are also differences between the two states at the extracellular side of the pore, which is shown here, above the selectivity filter. And these are also related to the position of this arginine. So we thought that we could discriminate between these two conformations with the nanobodies. We sequenced 100, and we found 10 unique nanobodies at the last step of retrieval, of course, of which we selected 5, indicated by the green arrow, because they were giving the highest expression level in Escherichia coli and also the highest stability of the protein once purified from Escherichia coli. And we started to check if they were specifically binding HCN4. We did this by fax, in which we had X cells expressing HCN4 and with the GFP, and the nanobody that was marked with the red Alexa fluorophore. So by doing this, by following the co-expression of the two colors, we were reducing the number to 2 nanobodies of our interest, because nanobody 5 and 34, they were showing some preference for HCN4 compared to binding to HCN2 and HCN1. So we also evaluated the affinity of these two nanobody to the isolated HCN4 protein, purified protein, by microscale thermophoresis. And we got KD, acceptable KD of nanomolar, 37 nanomolar for number 5, and 10 nanomolar, a little bit better for 34. Then we went on and we screened by patch clamp. We did a lot of screening inside, outside, but I'm showing you the data, the most significant data, which were obviously collected with the nanobody provided from the extracellular side, because this is what we are interested in screening for. And the best was nanobody 5, because nanobody 34 did not show any impact on the current, while nanobody 5, we found that is activating HCN4 current. You can see here that there is an increase of current when we add nanobody 5, but not in the maximal current, which means that this is a shift due to the shift in the activation curve. So it's affecting the voltage dependency of the channel by binding from extracellular side. And this is about 10 millivolts of right shift. It also has some impact on the kinetics. It accelerates the kinetics of activation, but is not significant, the change in the deactivation. We quantified the K1/2 by patch clamp of the nanobody, and it came out to be 40 nanomolar in our experiments, which is pretty in agreement with the one that we calculated by microscale thermophoresis on the purified protein. So we were surprised by this coincidence, of, let's say. We also retested, of course, that the nanobody 5 is HCN4 subtype specific, as the fax was indicating previously. And yes, it only acts on rabbit HCN4, but not on human HCN1 and mouse HCN2. And when we aligned the 3 sequences, we found several positions in which there is a HCN4 specific residue. So we were not sure of which of these extracellular loop was indeed the target of our nanobody. But then we were helped by realizing that the effect of nanobody 5 is also ortholog specific in HCN4. So it's not acting on all HCN4 protein that we had in the lab. We were testing human, mouse, rat, and others. And we realized, for instance, that nanobody 5 acts on human HCN4, but not on mouse HCN4. So we could restrict, by going back to the sequence, the region of interaction, and where rabbit, mouse, and human diverge. And they diverge in particular in these loops, which are related to the turret, which are connecting S5 to S6 or S5 to the P helix. And we found these are actually we restricted to 2 because these are the only 2 amino acids in which human and rabbit are identical, and only mouse diverge from them. And this is what we see in our functional data. So we started to we mutated, obviously, the 2 amino acids reverting to the mouse ones, And already the reversion of 1 gives half of the effect on the shift of the nanobody, and and both of them eliminate the effect of the nanobody. And these are the corresponding dose response curve. We go from 40 nanomolar to 400 with 1 mutation, and we go to basically non-responding with a double mutation. So the other thing that we needed to check before, let's say, going in ex vivo experiment was the possibility the nanobody 5 does not recognize the heteromeric channel HCN4, HCN1, because this is presumably forming in the sinoatrial node cells. And we tested, and in reality it does recognize the heteromeric channels, which means already that probably is acting only on the monomer and not at the interaction between two subunits, and you don't need 4 to activate the channel. At least you need 2. So we went to test the effect on the sinoatrial node myocytes. Of course, we tested the rabbit because we couldn't test the mouse, or we tested the mouse, but it was not working. And the current recorded, the IF current recorded from rabbit is indeed responding to the nanobody with a shift which is similar to the expected one, 12 millivolt to the right. This is the statistics. We noticed, we could measure an acceleration of the heart rate as expected by an increase in IF current. These cells shown here is this one that was going from 70 to 120. Others were responding. A few were not responding. So we have a statistics overall of about 15% of increase in heart rate when we add the nanobody. And this experiment was done in Mangoni's lab in Montpellier. We, in Milano, we tested the effect of nanobody 5 on mouse sinoatrial node. And as I mentioned, there is no effect as expected. So at this point, we are searching for a mutation in HCN4 that is causing in human sinus node dysfunctions. There are several patients in the literature that have been discovered. And one of these mutations that was published first in 2012 by this group is found in a familiar tachybrady syndrome. This patient has a mutation in the C-linker of HCN4 that is this K2 asparagine, and it has a left shift of 11 millivolt of the activation curve. So it tends to be bradycardic. And when we add our nanobody 5 to the extracellular site, we can fully recover the position of the activation curve back to the control condition. So this is promising, and it might be interesting to move on and to try to do in vivo experiments, at least in rabbit, or we have to find a way, because we cannot work on mouse. That's the problem. So my conclusions are that nanobody 5 is a HCN4 specific activator, modulates the native current IF in sinoatrial node myocytes, and we could use it to revert the effect of a human mutation that was found in a patient. And with this, I want to thank my group, Ati for the screening of the nanobodies, Andrea for the protein purification part, and Alessandra Roberta for the patch clamping experiments. And thank you very much for your attention.
Thank you very much for a great talk and for getting us back on time. So I'm going to ask a question very quickly. The effects of your nanobody 5 seem to be very similar to the effects of cyclic AMP. Do they synergize or do they oppose each other?
Yes, that's a very good question. They are additive. We were also surprised to find out that, as you see, the nanobody 5 shift to the right. This is the effect of cyclic AMP alone. And this is the effect of nanobody 5 plus cyclic AMP. They are mathematically additive. And it's interesting if I can say this, because of course cyclic AMP acts from the intracellular side to move the voltage sensor, and our nanobody moves from the extracellular side, but it's not binding to the voltage sensor, it's binding to the turret. So it's uncovering a non-canonical pathway that connects the voltage sensor to the pore, which is passing through the turret, that still is shifting to the right activation curve. So it has a lot of interesting aspects.
And have you looked at any effects of the auxiliary subunits, the KCNEs, that also associate with these?
That's great. I was wondering. I have to do it, yes.
Yes, Damien.
Come on, Jason, you're faster than that. You're a footballer. One day I was. Cheers. Lovely talk, Anna, and pun intended, an ion channel close to my heart. This nanobody 5, you obviously did these really nice HCN41 heteromer studies. Did you manage to do anything like concatenation where you might try and force it into a 3-1 or a 2-2 or a 1-3 format?
Not yet, but we have the concatamer because we have been publishing this many years ago. I have to look in the freezer. That's a good point. Okay. Thank you.
Take a good look in the freezer. There's some good stuff in there, apparently.
Any other questions? One last one there on this side. He's quicker now, Damien, when it's not you.
It is, yeah.
Very slick.
I'll take that to know.
It's isoform-specific movement.
So with HCN channels, you normally see like a pip depletion when you're recording them. Does NB5 have any effect on preventing that at all?
Can you repeat it? Sorry.
So you often see PIP2 depletion if you're recording with OSI.
A what?
PIP2.
Phosphoinositide.
Oh, of course, sorry.
Yeah. So
the rundown, you mean?
Yeah.
Okay, no, we wait for the rundown.
Oh, okay.
We always wait for the rundown, and then we start the experiment.
I didn't know if it was having an effect with maybe preventing the rundown or making
we
excluded the problem so far, but yes, that's another thing that we have to investigate. I have 2 things now to go home and do.
You have to look at your freezer for starters.
Well,
Okay, so thank you very much to all the first 3 speakers.